Okay, so good morning, everyone, and thank you again to the organizers for this opportunity to speak today at APBJC. I'm a research scientist from CSIRO in Australia, and today I'll be talking to you about a machine learning approach for identifying proteomic signatures of drug response in cancer. So proteomics is rapidly evolving as a really promising field for identifying predictive biomarkers in cancer. And this is because the proteome is thought to more reliably reflect the actual effectors in the cell, as opposed to something like transcriptomics. So to that end, the Wellcome Sanger Institute, in collaboration with our collaborators from Sydney, ProCan, recently published the world's largest pan-cancer proteomic map of 949 commonly used cancer cell lines, treated with 625 anti-cancer drugs. So this is a really valuable resource for identifying these predictive biomarkers. So what's really difficult to tease out, though, is that in cancer, there are often multiple misregulated pathways as a result of many dysregulation of proteins. So this effect is really hard to capture in the context of drug resistance and sensitivity. So we think that we should be able to capture these synergistic protein relationships. And so synergistic protein relationships in our study, essentially, we've just called non-additive effects, that where the combined effect is greater than the sum of its parts when contributing to drug susceptibility. And we think they may be more important to detect because they may have a greater effect on cancer cell viability than non-synergistic interactions. And this might expose protein hubs that connect important pathways within the cells that can help us to guide preclinical studies. So really, well-known example of a synergistic effect is synthetic lethality, whereby two orthologous pathways are co-inhibited to produce a lethal effect in cancer cells. So in a normal cell, you have two main pathways that fix double-strand breaks, and you have the base excision repair pathway, which is the PARP pathway, and you've also got the homologous recombination repair pathway mediated by BRCA. So in a lot of breast and ovarian cancers, it's actually very common to get BRCA mutations, which renders that BRCA pathway completely ineffective, which means that the cell is solely reliant on the PARP pathway for double-strand break repair. So if a patient has a tumor with the BRCA mutation, then if you treat them with a PARP inhibitor, then the BRCA mutant cells will be selectively killed because the normal cells have an intact BRCA pathway to compensate for that. So this is a really nice example of a digenic synthetic lethal effect, but there's actually a lot of evidence in the literature that suggests that most synthetic lethal effects might be complex and polygenic. So we think it's really important to identify these polygenic and complex synthetic lethal effects, and we think that and you can identify the more robust and penetrant effects contributing to drug susceptibility. So we think that a machine learning approach might be able to capture these nonlinear effects more effectively than traditional methods. So I apologize in advance, I don't have a fancy deep learning model for you, I have a random forest model. So we use Ranger, or our cloud-based implementation of the random forest model, which is called Variant Spark. And Variant Spark is geared towards using really ultra-high dimensional data. We've tested Variant Spark on a data set of 4 million SNPs, and about 100,000 samples, and it's the only tool that's able to run the random forest algorithm on such a big data set. So you can see that such a tool is really valuable for multi-omic data sets where the data set will be really wide and not as long, which means you don't have as many samples, but you've got a large number of features. So we tune our hyperparameters to improve the accuracy of the model, and then we use a simple method to identify synergies. And so this detects the higher order interactions and detects the protein combinations that positively correlate with IC50, which is the half maximal inhibitory concentration, meaning that the amount of drug that's required to kill 50% of the samples. So the higher the IC50, the more resistant the cell is to a certain drug. So without going into too much detail on how we identify and test these feature interactions, this is based on a hypothesis that was previously published, which saw that in a GWAS data set where you have lots of SNPs and complex phenotype, they found that SNPs that occur concurrently in the branch of a tree were more likely to be epistatic SNPs and contribute to the complex phenotype. we decided to extend this and essentially wanted to apply this to continuous data and multimodal data. And we hypothesized that this also applies to features such as protein or like genes. And so we think that the synergistic effects are more likely to be found in these concurrent branches. So we build regression trees. So we build about 5 to 10,000, depending on the data set. And the second step is to generate a count matrix of how often the pairs or triplets occur in the tree. And so the higher the number of counts, the more likely they are to be interacting. And then we fit a simple linear regression model. So we wanted to see if our method works on synthetic data, because we first have to establish whether this is even true. So we considered a couple of interaction models. I won't go into too much detail here, but essentially the ones we're really interested in are the synergistic ones, the interaction only, and no interaction. So we build some dummy data where we know the ground truth, we know which pairs or which proteins are interacting to produce the phenotype, and we've tested on a simple phenotype and a complex phenotype as well. So the first question we wanted to ask was, do the true interactions actually occur on top of that count table? So do they actually even occur with a higher frequency than others? So I apologize for the small text here. So on the x-axis, you have heritability, which is probably not the best word to use. It's more the variance explained by the model. So and then in the y-axis, you've got the median rank for that particular data set. So you can see here in yellow, which is the synergistic models, you can see that the median rank is often quite high, suggesting that if protein pairs do have a true interaction effect or synergistic effect, then they appear close to the top, if not the top, of that count table. So that's the first step to sort of reduce the search space and say, okay, we're going to test a certain number of interactions to see if they're significant. So I'll take this 70% variance explained data set to delve into a little bit deeper. So the next step is to see, is this interaction significantly different to the marginal effects of each feature? So the first observation I'll talk about is the synergistic interactions, and you can see that they are quite reliably identified, which is good to see. The redundant interactions, which means that the effects cancel each other out, were filtered out, which is also good to see. And this is a really important one. If the data set had no interactions, then we did indeed have no or very few results for that. Interaction only, again, was reliably identified, and dominant phenotypes were filtered out, which is good as well, because we don't want to keep the proteins that are fully explained by the marginal effect of one feature alone. So coming back to the actual data set that we've been working on, so the Procan-Sanger data set, we wanted to see, the main question we wanted to ask was, across all the 949 cell lines, which are across a broad range of cancers, are there any protein combinations that correlate with drug sensitivity and resistance? So using the random forest algorithm, we wanted to prioritize the protein groups to test for interactions. So keeping in mind this is 949 baseline cell line proteomes, so this is before drug treatment. So we wanted to see if the baseline proteome of the cells can predict how resistant or sensitive a cell line is. So this is an example volcano plot for AZD4877, which is a kinesin spindle protein inhibitor. And you'll see the resistance biomarkers in red. So the top right corner means that the coefficient is positive, which means that they're positively correlated with the IC50, therefore resistance biomarkers. And then you have the sensitivity biomarkers, which are in green with a negative coefficient. So what we did was, in the data set, we had 10 microtubule inhibitors. We took all the significant interactions for the microtubule inhibitors and just ran a gene ontology analysis. And it was pretty encouraging to see. We saw some gene sets consistent with the drug's mechanism of action, which included nucleokinesis and cell adhesion, which means pretty much nothing. But it was kind of like a sanity check to see what sort of proteins we're getting and whether we're completely off base. What we found interesting is when we sort of linked all the interactions together, we identified some interesting hubs that connected resistance modulators. For example, CD168 in the middle there is sort of central to two resistance-causing hubs. In terms of global drug class agnostic resistance biomarkers, which means it doesn't matter what the drug's mechanism of action is, are there any resistance biomarkers that we can see? So markers such as VAT-B and MCM4 commonly occurred across pretty much all drug classes, and pathway analysis suggests that cells with baseline higher proliferation rates and baseline higher endoplasmic reticulum stress are more likely to be resistant to a drug, regardless of what drug that is. And this is, again, nothing novel. It's pretty well established in the literature. Of course, we then had to replicate the hits to see if they validate. Unfortunately, there's not too many data sets out there that are that comprehensive, but we did find a data set from Damon et al., including some newer drug sensitivity data they've done, and the overlap was 30 compounds. So we found 183 common pairwise interactions, and this is keeping in mind that these 183 interactions are not just the presence of the pairwise interaction coming up as significant, but they have to also occur for the same drug. So we were pretty stringent in trying to identify which pairwise interactions actually overlapped with each other. Again, it was good to see that the pairwise interactions occurred in common groupings, such as EGFR inhibitors, so afatinib, erlotinib, and gefitinib, and a couple of other groupings as well that were not too clear. We found some consistently identified pairwise synergies in both data sets. Again, we found recurring pairwise interactions in certain groups. But it was also good to see that EGFR was commonly coming up for EGFR inhibitors, and this is after taking into account the marginal effect of EGFR inhibitor alone. What we also found encouraging to see was that Vimentin, or VIME in red at the bottom there, came up as a really, really strong resistance biomarker for, I think that's bosutinib and gefitinib, which are tyrosine kinase inhibitors. And it's commonly known in the clinic that if a patient stopped responding to EGFR inhibitor, you take a slice of their tumor and you do immunohistochemistry, you'll see high upregulation of Vimentin, which means that the cell has adopted to a more mesenchymal phenotype. And this suggests that, so it was pretty much resistant to the EGFR inhibitor. So the key take-home messages are that it's essential to define the molecular links between regulators of sensitivity and resistance, so we can identify the most penetrant effects. Our method is highly scalable. I haven't shown you a particularly big data set here, but it has the potential for multi-omic data that's super wide. So we think that the method may capture nonlinear links between features and can help us to guide preclinical studies. Of course, it requires extensive in vitro and in vivo validation. Some of the caveats include identification of higher order interactions, which prove to be a little bit more tricky, and this means triplets, quadruplets, anything greater than pairwise. We are testing different methods to reliably identify these. We're also testing different interaction models, as they might be data set dependent. We also do have an additional data set, and of course we're applying to multi-omic data, which I think is where the true strength of this method is. And with that, I'd like to thank my group lead and team lead and everyone else who was involved in the study, and our collaborators, ProCan, as well, and thank you all for listening. So any questions from them, please?
Yeah, beautiful talk. So quick question for you. In terms of personalized medicine perspective, we usually want to see whether some certain patient have the mutation before giving them a chemo, right? Mm-hmm. Have you ever done this kind of similar type of synergistic relationship between protein structure mutation and another protein expression? Yeah, thank you. For the drug response.
So you're saying like the actual mutation and the protein expression? Whether there's a... Yeah, no, we haven't actually done that, but that would be an interesting thing to look at for sure. We're looking at ways to also integrate some image data as well into the model. So I think that, that should probably be, the next step as well. One quick question? Please.
So in the SNP analysis part that you showed, I guess this was SNPs affecting the coding region of the corresponding genes. Mm-hmm. And if so, did you check whether the effect related to the predicted impact in the protein, whether this was loss or gain of function variants?
So we didn't actually use SNP data in this particular analysis. We are trying to. We are hitting into a little bit of trouble because you have the discrete data and then you've got the continuous data, and just normalizing them without adding artifacts is pretty tricky, but we're looking at different ways to integrate that at the moment.